welcome to the Wednesday afternoon lecture series coming to you today on a Tuesday. I'm Ela Serpe, and I lead the section on cellular communication in the Child Health and Human Development Institute. I'm thrilled to introduce today's speaker, Dr. Yishi Jin, who is conducting some of the most exciting research in synapse development and in neuron regeneration and repair. Our speaker today is Yishi Jin, a distinguished professor of neurobiology and cellular and molecular medicine at the University of California, San Diego. She has been nominated by the Women Scientist Advisors, first because of her science, and secondly because she mentored and served as a role model for many women, including myself. Jin received her undergraduate degree in cell biology from Peking University. She completed her PhD in molecular biology at UC Berkeley, working with Drosophila model system in Catherine Anderson's laboratory. Then she moved to MIT for a postdoc with Bob Horvitz, and there is where she started to use the nematode C. elegans as a model system. In 1996, Jin started her lab at UC Santa Cruz, and just 5 years later, she became a Hughes investigator, Howard Hughes Medical Institute investigator. Today, Jin is a distinguished professor and co-director of the Kavli Institute of Brain and Mind at UC San Diego. Jin's research focuses on the molecular genetics mechanisms underlying development and function of the nervous system. First, she pioneered the use of fluorescence reporter for in vivo visualization of synaptic structures. Then she used this tool to screen for molecules involved in synapse assembly and development. Her approach practically established the field of molecular genetics of synaptogenesis. In addition, she plays an instrument role in developing an in vivo laser axotomy technology that enabled our modern research on axon regeneration. Her lab not only discovered pioneer molecules in synapse assembly and or axon regeneration, but also elucidated their signaling networks during nervous system development and maintenance. For her science, Jin has received numerous awards, from Presidential Early Career Award to Fellow of American Society of Arts and Sciences, Fellow of the American Society of Cell Biology, and last month, member of the National Academy of Science. Jin is among the most highly cited researchers in the United States. She is also dedicated to mentorship and science education. She has trained dozens of young scientists, many of whom are now in tenured or tenure-track position all over the world. The title of her talk today is Neuronal Response to Injury and Stress, Insights from C. elegans. Jin, welcome to the NIH. Thank you so much for being with us today.
Thank you very much, Ella, for that very generous introduction, and also thank you for WALS to nominate me for this lecture, and it's really humbling to speak here, and particularly appreciate all these years the NIH support to my research program. So today I will, as the title implies, and then I will basically cover from the work we use C. elegans as a model to understand the fundamental mechanisms of how neurons protect themselves. But I will start with, let's see, is it moving? No, going. I'll start with this century-old image. There's quite plenty of them you see at the NIH campus that was produced by Ramon Cajal, where through very systematic, extensive microscopy observations, I'm just trying to figure it out. This is a laser. Is that right? Oh, which one's laser? Oh, it doesn't work. So I'm supposed to be using the laser on my screen. That work? Okay. Right, okay, so I think you can see that. That is through a lot of work himself and his students, and they demonstrated not only the neuron doctrine and later on and explored the concept of how neurons, they're post-mitotic, how do they respond to injury. And this image actually shows the dogma that is in the central nervous system in responding to a knife cut. All the axons looks still there, but it's very unhealthy with the end bulb, and it's looking as a death sign, or so-called dystrophic bulb. And it was the dystrophic bulb and had triggered a lot of studies over century, and some contributed to extracellular environment such as myelin inhibiting factors, and some contributed to intracellular disorganization of a cytoskeleton, such as this study that was done by Frank Bradker's lab, which shows disorganized microtubules. And many efforts then have been evolved over the years, both through inhibiting glial scars inhibitors, or through microtubule-based stabilization, and to help injured axons and to protect the severed ends to regenerating. But despite all this many years' efforts, our understanding about how mature neurons responding to injury remain to be an intense topic with many theories. So I have been working with model organisms more or less for 20, 30 years, particularly using nematode C. elegans as shown here. It's transparent. The nervous system has a fixed number of neurons, 302 of them that are fully reconstructed through the work of John White and others. We know exactly where those 300 neurons are located, and we can visualize every single one of them using transgenic fluorescent marker. And in C. elegans, axons grow along conserved extracellular matrix, and they're not myelinated. Generally, their morphology is rather simple. In many studies, probably serendipitous ways, we collaborated with applied physicist Fatih Yannick, and to initiate using laser as a way to sever the C. elegans axon. And here shows a movie that essentially you'll see this is a laser cut, and then this is a compiled about 7 hours movie, where you see is the severed axons and begin to initiate growth cones and filopodia to extend, very spontaneous, very rapid. But this regrowth is actually, it's very spontaneous. It doesn't actually repair themselves and completely back to its developmental state. So when we established laser axotomy assay, we decided to take advantage of C. elegans single cell and transgene applications, and to decide to probe into the underlying mechanism. And the first thing, as we all know, generally calcium is the first responder to a lot of traumatic injuries. So we used, this is early studies about a decade ago, we expressed the GCAMP for calcium sensor in the C. elegans neurons. As you see here, following injury, laser severing, you see the transient of calcium as a rise of orange color and bifurcated towards both distal axons and the soma. This is also very spontaneous, but we can measure the transient amplitude at cut site to total growth extent, where we can see a positive correlation, and suggesting the calcium transient comes with the injury, likely contribute to injured axon to regrow. Indeed, through genetics pharmacology, we're able to link the calcium transient to voltage-gated calcium channel as well as intracellular store. And this calcium, very rapid calcium transient, works together with cyclin P second messenger system to promote axon regrowth. In many ways, that cyclin P signaling promotes injured peripheral nerve to regenerate, has long been studied in the mammalian field, particularly Wolf and also Maria Philbin many years ago. And more recently, we identified this rapid axonal calcium transient is followed by a slower calcium transient in mitochondria, which also depended on mito unipolar, which is balancing the cytoplasmic mitochondria transient. Additionally, we've been exploring both membrane organelles as well as the cytoskeleton. And one of the very traditional marker to looking at cytoskeleton is a microtubule plus unbinding factor. It's called the EBP. So what you see here is a single axon, and the axon is less than 1 micron diameter, and there's probably only about 10 or 15 microtubule fibers. And in this movie, you see injury site, and the EBGFP forms a bright white spots, which converted to the chemograph, and the EBP site correlating to the microtubule dynamics that induced by axon injury. So in uninjured axons, there hardly see EB plus comet. And within 3 hours, this is the time we start seeing signs of philopodia, and there are increase of EB plus number. And by 6 hours, and those severed axons get stabilized, begin to elongate. In both cases, we start seeing the EBGFP comics, the EB comics first increase and then sustain the growth in running lengths. Additionally, we identified a molecule, we call them EFA6, which is a microtubule destabilizing factor, responding to injury translocate to microtubule minus end, and then that triggers destabilization microtubule, and to work together with 2 conserved protein called TACC and then double coating kinase. This finding subsequently followed by James Fass's group to characterize the mammalian EFA6, which also show in peripheral nerve, mammalian EFA6 acts as axon regeneration inhibitors, in part by coordinating with microtubules and organelle transport. So our main goal, by setting up this laser axotomy assay, is to take a functional gene conservation screen to look for mechanism could be shared between C. elegans axon, which is very different morphologically with the mammalian axon regeneration field. So the way we did it is that C. elegans generally encodes about 21,000 protein coding genes. A third of them has been characterized as direct orthologs between C. elegans and human. We have wonderful resource community that generated lots of genetic knockout mutations. And so we then start choosing about 1,500 of them that we know could be expressed in the nervous system and could be influencing axon regeneration. In all these cases, we choose the genes are homozygote viable and generally with a normal nervous system architecture. Then we perform axotomy in late larvae to adult stage and to look for axon regeneration response in a way it's more resembling the mammalian regeneration field. As shown here, essentially we screened close to all those 1,500 genes. And in fact, the majority of them, almost 80% of them, have no defects. What I mean by that is shown in this image, that in responding to injury as this red arrow, and those axons will initially regrow, but you can see here it's grow randomly over the place. And this error-prone regrowth is representing a spontaneous response to injury. We see about 15% of genes that loss of function in those genes resulting decreased regrowth, and that means the gene normally is to promote injury response. And as you see here, the image is basically there's not much regrowth, there's very little branching. About 5% of genes ended up acting as axon regeneration inhibitors because they're more exuberant growth or total growth is being more compared to wild type. Because these genes are chosen based on homology between C. elegans and the human, of course, those genes then fall into all categories from gene expression and to signaling molecules, trafficking to extracellular matrix molecules. And this summary just shows is that within each gene goal categories, and there will be genes required for promoting injured axon to grow, there are also genes required or acting to inhibit the axon regrowth. So in many ways, as a geneticist, this feels very fitting. That is, axon regeneration following injury is a process that somehow mimics a developmental forming of the whole nervous system. It requires combinatory actions of a variety of genes. And there's unlikely, and we often say, there's a single master regulator to control axon regeneration, whether you repair yourself or not. So with this information in mind, and we start taking a molecular geneticist approach, is try to put the genes and their actions into signaling pathway. And this is basically by systematically characterizing the key genes and expressing those genes' molecular gene function, and plus in vivo imaging. And so in generally, we divide the axon injury response into 3 stages. A very immediate stage is this calcium transient and instant, which then these followed with the initiation phase of axon regrowth. This basically involving membrane repair, cytoskeleton reorganization, initiation of growth cones, which then followed with extension. This extension phase can grow more or grow less. And what we find is our focus today mostly on the DLK kinase, which is stress sensing kinase, and a bit about a RNA protein binding factor mediated phase separation. And then later on, I will talk about the translating C. elegans finding into mammals, and then we'll come back to DLK, how we currently understanding its in vivo regulation. And this is showing here is in the later stage during axon elongation, all the developmental guidance molecules start coming into play to modulate the growth direction and growth extent. So let me start with this protein called the TIAR-2, which is an RNA binding factor, typical standard RNA binding factor with the 3 RNA binding domains. It's conserved from C. elegans to human. It's named as a TIA is simply because it's a protein expressed a T cell as an antigen. And what's characterized this TIA family is besides the 3 RM domains, it has this prion-like domain, which tend to form some sort of aggregates in various proteins. A few years ago, there were reports about point mutations in the human TIA1 genes are actually linked to stress granule dynamics as well as ALS patients. Matt Andrusik, a former postdoc, basically characterized a lot of RNA binding factors and found this one that is loss of TIAR-2, resulting more extended regrowth after injury, which is quantified here. So we normalize the regrowth wild type as 1. So anything above 1 with a statistic significance shows that losing TIAR-2 and leads more regrowth extent. Additionally, Matt did a time course analysis to show TIAR-2 actually acts in the early phase in the growth cone initiation. He then did transgenic rescue by putting TIA into the neurons, which fully rescue the extended regrowth phenotype, but this doesn't happen if they're putting the genes into muscle cells. Additionally, he can place the human TIA and into C. elegans neurons, the TIAR-2 null mutants, which also fully rescue the TIA null mutant phenotype. And as you see here, when he increased the expression level of TIAR-2, he starts seeing inhibition effect, that is, there's a reduced axon regrowth. So TIAR protein behaves as a dosage-dependent factors influencing axon regrowth. And the TIAR-2 protein also exhibiting a pattern that's also dependent on its dosage. In some cases, in the axons, in this single neuron called mechanosensory neuron called PLM, and high expression level will form granules, and low expression level is rather diffuse. This can be quantitated here. This kind of a concentration-dependent granule formation, and right around the time, and came with this idea of liquid protein phase separation through liquid-like droplets behavior. And as Matt then purified the TIAR-2 protein, and as well as GFP tagged the TIAR-2 protein, performed the liquid droplet assay, which basically has crowding factors, where he instantly saw that there's a formation of droplets. Those droplets, one of the behavior is that they have liquid behavior, is when he frapped them and they recover very rapidly. And this TIAR-2 axonal granules is actually very, although in cell cultures, TIA protein has been considered as a core components of stress granules induced by stress. But in C. elegans axons, we did extensive co-localization studies with other RNA granule factors, synaptic vesicle proteins, and synaptic actosome proteins, as well as mitochondria. As you see here, is except TIAR-2 itself, co-localized with itself, generally there's very little co-localization, indicating that axonal TIAR-2 protein is rather heterogeneous. It's very dynamic. And we see this dynamic aspect of it in this live imaging, and that is where you see the TIAR-2 granules will basically coming in fuse and split, but they actually don't now go long-range transport like synaptic vesicles. They also responding to injury, this dynamics split and merge as shown here. Pre-injury, there's one cluster, and after injury, and then this triggers sometimes fewer, sometimes more. But this is a very transient event. Then Matt decided to address the mechanism, what drives this TIAR-2 behavior. For this, this basically summarized his 2 years' work, essentially expressing GFP-tagged TIAR-2 in vivo in a concentration very nicely controlled way, and then taking out each domains and purify the proteins and to assay its in vitro droplet behaviors, as well as in vivo whether it forms axonal granules or not. And on top of that, and to show if it can complement or rescue the loss of TIAR-2 axon regeneration phenotype. And in snapshot is that the full-length TIAR-2 forms axonal granule and then rescues the enhanced axon regrowth of TIAR-2 null. And when he deletes all the RNA binding domains and there's no, the axon granules, when he deletes all the RM domains, axon granules still form, and then the activity of rescue the mutant is still there. But when he perturbed the prion-like domain, just even small domain, which abolished the axon granules and also abolished its a functional activity. So really is this small domain that can nucleus granules, it's what provided the TIAR-2 function. And I'm not going to go into additional details. He was able to identify there's a phosphorylation events, both the serine tyrosine phosphorylation in that small domain. It's responsible to trigger this phase separation. So we came up with this model that is in axons, TIAR-2 proteins are always there, and they undergo a very dynamic liquid-liquid phase separation, which can be modulated by ion balance and injury-induced changes, as well as phosphorylation. This the temporal granule formation might be is a stage that sampling injury signal somehow spread that signal into the entire axon. So this study was about the same time very nicely synergized with Jeff Twist's work using mouse sciatic nerve, where they identify a canonical binding factor of TIA protein called the G3BP1, also undergoes formless granules in a phosphorylation-dependent manner. So appear to be that perhaps liquid-liquid phase separation in axons could be a conserved mechanism from C. elegans to mammals, perhaps to human as well. So this stage, that's how we dissecting the protein of TIAR-2, and I'm just going to transition to my favorite molecule for some long time. It's this kinase called the DLK-I. And the DLK-I named is because it's a MAP3 MAP3K kinase called the do-losing zipper kinase. It's not entirely accurate in the sense is in mouse and in human, there are actually two sister kinases. They all characterized by two losing zipper domains with an N-terminal kinase. One is called MAP3K12, one is called MAP313, and happen to be the K12 is given name the do-losing zipper kinase, whereas the 13 is called the losing zipper kinase. Whereas the invertebrates, Drosophila and C. elegans, all have one, and Drosophila one's initially named as a Walenda. C. elegans has a simple name called the DLK-I. Apart from this conservations, all of these kinases actually subjecting to palmitoylation-regulated lipid modification. We studied the DLK actually long before we established the laser axotomy assay. We studied the DLK from the early interest of my lab, that is synaptogenesis, how do you form synapses. We used synaptovesicle labeled GFP to mark individual synaptic terminal. We did a genetic screen to looking for mutants disrupting this very nice synaptic puncture. And this led to a gene called RPM1 because as a regulator presynaptic morphology 1, because this cluster becomes very irregular compared to wild-type normal-shaped puncture. Very nice organization with vesicle cluster surrounds an active zone protein called Liprin SID2, whereas disorganized synaptic cluster in RPM1 mutants just have a large spread vesicles with multiple active zone. And turns out this RPM1 mutants is RPM1 protein. I think this feels like my voice is not transmitting. It's green light anyway. So it turns out the RPM1 protein is a founding member of a conserved protein family together called the PHR, because human protein is PAM, and then the Drosophila protein called HiWire. And these are large proteins that's close to about 4,000 amino acids, more or less. And they all, over the decade, from the work of Corey Goodman, Aaron D'Antonio, Cassie Collins, Sam Pfaff, and Rob Burgess, all come together from genetics and molecular genetics approaches to show that every single member of this protein plays important roles in different stages of axon termination and axon guidance and synaptogenesis and synapse stabilization. A common feature of this PHR protein is that this ring finger domain, and suggesting it is actually an E3 ubiquitin ligase. So now this raises the question, is an E3 ubiquitin ligase acts at the synapses, and so what will be its substrate at the synapses? So for this, my lab took a genetic approach, is to look for suppressors of RPM1 synaptic phenotype. The simple rationale is if the E3 ligase is not there, some protein must be upregulated, its signaling pathway is upregulated, then you can knock it down and then could restore the signaling homeostasis and then getting the suppressor mutation. This turned out to be a very effective suppressor mutation. We identified a MAP kinase cascade with C. elegans DLK-1 as upstream kinase, MAP triple K, and then with 2 downstream kinase as well as kinase, and then finally a transcription factor of the BZ domain transcription factor. That's knocking out each one of them will suppress the synaptic defects. And we have further showed the DLK kinase itself biochemically is a substrate of RPM1E3 ubiquitin ligase. And you could see here in vivo, this is the C. elegans neuron, and DLK is very low in abundance. Remove RPM1, you see its abundance is increased. All of these are really beautiful genetics, and we enjoy it very much. But then there's a question coming in. It appears to be during development, in the initial formation of synapses, that DLK kinase needs to be rapidly turned over to make sure the synapses are formed properly. But loss of function of DLK and any of its downstream targets actually is not essential. That's how we can find them as a genetic suppressor screen. So this sort of raised the question, what is really the important function of these genes, even though seems to be, its importance is just to be degraded, activated, it gets degraded. Until we made the connection when we're doing the laser axotomy screen, and then we rediscovered the DLK, and to be absolutely essential to initiate injury-induced regrowth, as you see here. Compared to wild type, and there are 10 or 12 hours, there are growth cones, the axon elongate, and loss of DLK, and the growth cone don't form, and the axons stop growing. And that's quantitated here. If we elevate DLK, and that resulted overall increased regrowth, and that increased regrowth completely dependent on its targets, such as this transcription factor. By itself, they do not regenerate, and also it blocks the enhanced DLK overexpression effect. Besides triggering this developmental signaling cascade, DLK also promotes injury-induced microtubule dynamics. If you recall, when we used the microtubule plus and binding factors, and we observed that there's injury-induced EBP comics and sustained EBP growth. In DLK loss of function mutants, both are reduced or blocked. There's fewer comics gets induced, and the sustained growth doesn't happen. Additionally, by combining with other genes, we could show part of this is due to disruptions in microtubule post-translation modification, such as tyrosinated tubulins. So at this stage, there's a lot of interest in now linking this invertebrate DLK. Both my group work on DLK-1, and Aaron D'Antonio and Cassie Collins work on the Drosophila DLK. This then led us to start asking what happens in the vertebrate case. As I introduced to you, that vertebrate actually has 2 homologs, the DLK and LZK. In fact, both kinases are expressed in the nervous system, cloned from cDNAs made from brain. In culture cells or cell lines, both kinases can activate the junk signaling, OP38 kinase. And there are multiple DLK knockout mice, and generally constitutive knockout die perinatally. Nervous system pretty much normal with subtle defects, but if it's deleted in adults, this is the work of genetics and neuroscience departments done, and those DLK mice perfectly normal, living as adults, and more or less resembling the C. elegans and Drosophila knockout. In contrast, the mouse LZK was very little study. We made a constitutive knockout. Again, it's perfectly viable and has no any detectable defects. But there's, in the last 10 years, indeed, from using mammalian axon regeneration researchers, and particularly from optic nerve injuries, this is from Genetech group, and as well as the Johns Hopkins group, and some people using the peripheral nerve injury and with R&D Antonio, have all shown is that the DLK kinase is important for optic nerve injury induced axon sprouting, optic nerve injury induced RGC neuron survival, as well as a peripheral nerve regeneration effect. My lab, and in collaboration with Yishi Jin's group at UCSD, and then decided to focus on the LZK kinase, where we show that LZK has a stronger role in astrocytes following injury, and this is now called astrocytes boulder, and that induced by injury is at least involving LZK. I know Claire Lepchon's group at NIH have also implicated DLK's role in microglia, and her early work, and particularly was important at the Genentech implicating the DLK kinase, is protective in animal models for neurodegenerative diseases such as Alzheimer's and ALS. In recent years, through collaboration with Yishi Jin's group, we addressed the fact, do DLK and LZK have any role in mouse spinal cord axon injury? And in this case, it's really extensive mouse genetics, and Yishi Jin's group have to use the P10 knockout as a background, because in the spinal cord, which is the central nerve system, and axons don't regrow unless you're knocking out the P10, they start sprouting. And so that was quite a lot of work, and what in this figure is too much to look, but just important thing to look at this. This is a control where there's essentially following dorsal hemitransection, and there's no sprouting of the CST axons to cross the wound site. P10 knockout, it will have more to sprout, but removing DLK and LZK, essentially, there's very little sprouting. It's the cleanest when to block the P10 effect. Individually, DLK, LZK do not have this strong effect. So DLK and LZK acting synergistically to block spinal cord axon regeneration and induced by T10 deletion. This is all good to support that the whole conserved DLK kinase, and along with the sister kinase, are important for injury response from C. elegans to fly to mammal. But at the same time, you really have to think about it's not just to block things. You have to think about active DLK so that you can promote axons to repair, to regrow, and to do something better. In this case, it was interesting to mention these two studies. One is this work from UCLA group, and showed that in a particular condition and stroke model, and inhibiting CCR5, which is a cytokine receptor, and somehow helped the mouse to recover from stroke. And this is due to the fact that they were able to show by blocking CCR5 and following stroke, and DLK is upregulated, and which triggered CRAB signaling, which triggered motor cortex neuron sprouting. And in some patients who have received CCR5 inhibitors, in fact, also recover well. So that is to say, in this CCR5 stroke model, activating DLK indeed help the motor axon plasticity to help recover from stroke. But on the other hand, there's also this study from Tom Sudov's group, which addresses using human-induced stem cells and addressing how human cell-induced neurons responding to ApoE factors, which is a major trigger for Alzheimer's. And where they show is by applying the ApoE4, and the cells will respond very potently upregulating DLK kinase. And the downstream of that DLK kinase is a transcription upregulation of APP. In other ways, you see is that elevating DLK can have, depending on what a system you're setting up, can be good to promote axon sprouting in stroke model, and in some ways it's bad to promote APP production, which also consistent with Claire's work that is inhibiting DLK can reduce neurodegenerative phenotypes in APP mice model. So by this stage, my lab decided to venture into mouse models. In this case is two brave people in the lab. One is a postdoc Yunbo Li, one is a graduate student Aaron Ritchie. So they decided to both examine DLK and LZK knockout mice as well as induced overexpression. But they focus on the cerebellum, the Purkinje cells, because we have our colleagues to help us learn the mouse anatomy. And suffice to say, after made a conditional knockout of DLK and NLZK and a double, and basically the cerebellum developed quite normal. Purkinje cells are very happy. There's no any defects we can detect. Then Yunbo and Aaron used this, we generate the transgenic DLK that's a crate dependent, allow us to manipulate cell type. When we cross them into PV crate, and which is active after P5 and P7, in this case, DLK is upregulated about 2 to 4-fold, which actually we could see, I'm now showing the data, active junk signaling, in fact, induces caspase pathway. And most of the Purkinje neurons basically rapidly die, and by P21, those mice also die, presumably due to its affecting other neurons, parvalbumin neurons in the brain. In contrast, we made the same mice, same kind of mice, but this time is inducible overexpressing LZK and cross to the PV crate. With those mice quite happy live up to a year, we see a very progressive, slow degeneration as you quantitate here. They can live up to 4 months. It's about 40%, 60% death. And LZK overexpression also induced the junk signaling as the phospho-C-jun target signal, and as well induced the caspase. So this is where we start asking the question, how can these two kinases and cell lines seems to behave the same, but in vivo, when we overexpress them in the same type of neurons, they appear to have different potency and in neuronal survival. And a lot of this is a generic thing. Kinases are triple kinase will activate the double kinase and then going through the junk signaling, so stress kinase. In thinking about the two MAP kinase differences, and we read this paper, which actually study T cell activation. In this process, junk signaling is widespread. And one of the junk signaling is induce many genes that's involved in T cell differentiation. And in this case, this group actually identified a mechanism is to show that the junk upstream kinase, MKK7, is undergo a alternative splicing by a RNA binding factor. It's called CELF2. Essentially is that the mouse MKK7 has an alternative splicing in a kinase docking site. The presence of CELF2 splicing factor will exclude this alternative exon, which then generate an active double kinase. And removing CELF2 then will result in a less active MKK7. So what we happen to realize is that there is a C. elegans homologous CELF2, it's called UNC75. And the CELF2 is basically a RN binding factors with the 3 RM domains. It's mostly nuclear, and it's named as TUD binding factor as LV specific from Drosophila genes closest Bruno. And the work of my former postdoc Li Zhenchen, who's now tenured professor at UT Health Science, actually characterized the UNC75 mutants and in axon regeneration, where she observed that losing on 75 and the axons basically don't grow much. Not as severely impaired as a DLK loss of function, but definitely quite reduced the regrowth. She's also decided to venture into mouse field and then got the CELF2 conditional knockout and then looked into synaptic nerve from the DRG axon regeneration. This is quite standard. That is, she basically conditional deleting CELF2 from the DRG neurons as marked by the red, and that's again PV-CRE. And the regenerating axon following the crash in the sciatic nerve is marked by ICG10, which is also Stephanin 2. So in the wild type, following this crash, and there will be peripheral nerve would regenerate and regenerate quite well, whereas losing CELF2 and there's reduced regrowth. And in combination with ECLIP to looking for splicing targets, Legion then identify a series of targets of CELF2, which included synapse transmission genes called syntaxin. So, but now we can start putting together the C. elegans work and the mouse work on CELF2 to ask the question, could CELF2 then influencing the DLK and LZK effect through alternative splicing of MKK7? And for this, we basically made extensive genetic mouse generate the combinatory mouse combinations, and the conditional deleting CELF2 in the DLK overexpression mice, and then asking, do they block the Purkinje neuron cell death? With DLK overexpression, and there's no effect whatsoever. But with LZK overexpression, we see a very significant suppression of LZK-induced Purkinje neuron death. And those animals are living much longer and happier. So through this cross-examining signalings from C. elegans to mammals, at least we're feeling one part of the knowledge in the Purkinje neurons that DLK probably acting through a different MAP kinase called MKK4 and then trigger a rapid junk signaling and caspase-mediated process, whereas LZK acts through a MKK7 MAP kinase and then trigger a slow junk signaling and then leading to Purkinje neuron death. So that says both kinases, even though in cell culture they behave the same, but in neurons they seem to have neuron type specificity depending on their downstream signaling. And as we were doing all of this, and then you probably say this is all published work and it's all well understood, but you still haven't answered the question, even in C. elegans, how is DLK level regulated? So I'm going back to my roots, working C. elegans and do genetic strains. This is the work of a graduate student, Yue Sun, and postdoc Junxiang Zhou. So what they did is they took advantage of genetic tagging endogenous DLK, and then they decided to look for strains of what could be upregulating DLK, right? So what we're showing here, this is a C. elegans worm, and the nerve brain, the whole brain is more or less here with lots of soma, and then the neuropils are connected here. And when we see endogenous DLK, and there's very little fluorescence you can detect. I can see on screen, I don't know whether that will show up. Then they feed the worm with mutagen, and they isolate mutants that will increase DLK level. In some ways, like the transgenic mice, but we're looking for endogenous mutation. Sure enough, we got our positive control because DLK is not only inhibited, degraded by the E3 ubiquitin ligase RPM1, we also identify a tubulin, and this is unexpected, and this tubulin is called BEN1. The C. elegans BEN1 tubulin is quite well studied, actually was studied for some years from Marty Chelfie's group and Monica Driscoll. BEN1 is a neuron-specific beta-tubulin. It was isolated because worms are sensitive to this microtubule drug called benamin, and grow on benamin, they eventually become sterile, they die. Loss of function of BEN1 resistant to benamin, and they grow on drug, they're perfectly fine. And then, whereas our mutation is this one in the beta tubulin, we model them, is in tubulins are GTPases, particular beta tubulins is pretty active GTPases, and they fold extensively, and there's this loop, it's called the T7 loop. It's actually in the alpha beta tubulin dimer interface. Our mutations affecting this leucine residue in this T7 loop behaves as a gain of function because it's hypersensitive to benamin. Now, the BEN1 gene may be hard to remember, but it's easy to make for those who are familiar with the vertebrate tubulins. There's always many different isotypes. The vertebrate type ones is TUBB3, which often is the same as what the TUG1 antigen antibody recognize. And there's been many point mutations disrupting human TUBB3 that resulting in neurological disorders. And BEN1 is basically is also log of mammalian TUBB3 because when mammalian TUBB3 expressed in yeast also confers sensitivity to benamin. What this gain of function mutation, what I showed you, is isolated because it's upregulated DLK level. Not only upregulates level, it also activates its downstream signaling. And if you recall a little bit, it's downstream of DLK signaling is this transcription factor, CEBP1. When we tag the endogenous CEBP1, it's ubiquitously expressed in all tissues, and it's very low in the neurons, in the head and most places. But in the BEN1 gain of function mutants, now you can see its expression level is upregulated in most of the neurons, not in non-neuronal tissues, and we can quantify them here. And this BEN1 gain of function mutation also acting in parallel to RPM1 loss of function, where we basically made the double mutants, the wild-type worm, as a real worm moves well, and sinusoidal pattern. BEN1 gain of function pretty much normal. RPM1 loss of function also pretty much normal. But when we make this double mutants, they're live, but they're paralyzed. When we're looking at the synapses, you can see here the synaptic puncture in the wild type. It's very discrete. But when gain of function, it's pretty well discrete. RPM1 loss function disrupted the shape of synapses, and this double mutants basically loses most of the synapses, and the mouse is paralyzed. So BEN1 gain of function acts in a parallel pathway, and since it is a tubulin, and naturally we looked into microtubule dynamics using the EBV binding assay, doing spinning disk, and then to generate the chemograph. This particular BEN1 gain of function, actually the microtubule dynamics is only subtly altered. It's not dramatic. And what we found is this BEN1 gain of function subtly changing microtubule dynamics and activates DLK moderately, actually has a protective role in synapses in motor neuron function. And this is shown by looking at the synaptic puncture, that wild type and the BEN1 gain of function, DLK loss function all look fairly similar. But the reason we say DLK, activated DLK by BEN1 gain of function is protecting synapses is then we take out DLK in the BEN1 gain of function mutants, the synapses gets severely disrupted. This is the opposite if I say that when we did RPM1 loss of function and taking out the DLK loss of function, we restore synapses. But in BEN1 gain of function, we take out the DLK, and we actually make the synapses worse, the motor behavior worse. This is a moderate increase of DLK level. When we treat the animals with spenamine, which is basically depolarize many tubulins, this is very dramatic. When we treat this, and DLK is dramatically upregulated, and the microtubule dynamics is severely altered, both in the number of tracks you detect as well as very short running lengths. In this case, what we see is that spenamine treatment depolarized majority of microtubules. It resulted in severe synapse defects, as you see here. This is wild-type worm without drug. This is wild-type worm with a drug, and there's severe reduced synapses, and animals were paralyzed. And DLK actually blocks this effect. So in the severe case, DLK is now is basically can block the microtubule disruption, induce the synaptic disruption. And in this case, we see the kind of a dosage effect, that is modest disruption of microtubules, and this is my last slide. In the steady state, DLK signaling is moderately activated, degraded, and tightly regulated by E3 ubiquitin ligase, and so that the signaling constantly under control. With the BEN1 gain of function, and there's a slightly changes in microtubule dynamics, which upregulates DLK signaling slightly, alters the signaling. The purpose of upregulating the DLK signaling is to counter the changes of microtubule dynamics to keep the synapses back to normal. But when microtubules are severely disrupted, in this case, DLK is way upregulated. In this case, everything exceeded the threshold, and synapses just becomes totally unstable, and neurons no longer function normally. So that leaves me with my working model from starting from one screen to another screen and roundabout testing our hypothesis using mouse models. We'd like to think is that this stress kinase DLK, its activation is tightly coupled to microtubule dynamics. In a way, is associated with neuron-specific tubulin isoform, and how this tight coupling is actually mechanistically what level is the question mark, we do not know. But I think what it is depending on the strength of its activation, and it will create a context-dependent outcome, whether neurons will survive, form normal synapses, regenerating or degenerating. So we have a lot more work to do, and I just wanted to end the talk by sharing the people in my lab and the first outings after last year pandemic, and the key people who did the work are Aaron, Richie, and Yue Sun, Junxiang Zhou, and Yunbo Li, and my collaborator UCSD, Andrew Chisholm, in the elegans axotomy screen, and Binhai Zhen in collaboration in the spinal cord injury. Our past lab members, Li Zhen Chen and Kei Kim, Matt Andrusik, all contributed to each of the stories I talked to you about. Thank you to NIH for very long sustained support, and Craig Nelson helped me to set up the mouse work. Junior Sayo, who's a hero in the football era, and for which I have the honor to be endowed chair, and the rest of the supplies, endless of strengths. Thank you very much for coming.
Well, anyone is free to use the microphones to ask a question. I have one that's come in online, if you're ready for it. It's a two-part question. I'm going to see if I can get this down. Thank you. This is from Bo-Mi Sung. He's a researcher here. Thank you so much for sharing this awesome work. Very interesting. You focused on injury-induced regeneration in axons. Is the same mechanism employed for damaged dendrites? Part 2. does the injury-induced regeneration fully restore function of the damaged axon to the state before the injury? Axons carry many functional units, for instance, axon initiation segment, axonic synapses, and acquired synaptic strength. Thank you so much for answering this question.
Well, thank you for the question. The first question is easy to answer. I did focus on, in all my our work, focus on axon regeneration, but one thing keep in mind, the C. elegans are simple axons. Some of those axons are mixed polarity. We have in our lab, working with the C. elegans, we have tested multiple type of neurites, including purely dendrites. And it's interesting, dendrites in C. elegans generally don't regenerate. It's not dependent on DLK. And actually, it's not because DLK is not there. It's just they don't regenerate. I will talk about, offer probably people know, whereas in Drosophila periphery neurons, dendrites do regenerate, and those process has been extensively studied from the Jan's lab and Yuan Yuan Su's other people's work. So in that sense, I just simply say it's because C. elegans axons are have unique architecture. We focus on axon regeneration. But second question. Axon regeneration is very complex. I totally agree. And at this stage, from the C. elegans work, we said our goal is look for genes that will respond to injury. So in that sense, we have not explored into in-depth of different forms of whether axon will sprout, will branch, form synapses, and in any way. But in collaboration with Binhai Zhen's lab, we have actually, in that paper we mentioned, we looked into spinal cord injury, which has, when they do dorsal hemitransection assay, the corticospinal cord can have multiple things. One is there will be spared axon and sprouting. There are also basically completely uninjured axons also sprouting. And what Binhai Zhen's group has observed that both of those effects are dependent on DLK. So there is a signaling events that somehow neurons will sense injury is present and they will respond. In all cases, I've not seen the reforming synapses by manipulating single genetic factor. Sometimes using multiple factors can make the injured axons to grow better, but the synapses is not formed properly. Although in C. elegans, some of the work we know is that they can be misconnected, therefore can regain certain degree function depending on the circuit, such as if it's the ultimate synapses is innervating muscles.
Thank you very much. It's been quite a marathon of interesting stories. My question has to do with that prion domain of the TIAR-2 that is required for both function and droplet distribution. You described that phosphorylation of that domain, which in my mind is promoting aggregation, or it's prone to aggregation. And also in somebody else's group, phosphorylation enhanced that partition. Isn't that counterintuitive?
Yeah. So there's two types. Phosphorylation, how it's modulating, and sometimes it could be it's triggering signalings, but phosphorylation also change charge environment. Mm-hmm. And so the phase separation has a lot to do with the crowding, how you get to your neighbors. And I have seen sometimes reporting phosphorylation promotes droplet formation, sometimes phosphorylation, and that just push the droplets to solid state. And I think a lot of this coming probably is eventually discover who is forming droplet with.
I see.
So that's at this stage. We haven't actually identified the kinase except when we made the phosphomimetics and mutate those serine residues. We see changes. Is by extension, is phosphorylations involved.
Thank you.
Maybe concluding comments, Dr. Serpa?
Thank you so much. It's been a pleasure to see all of this together. We all have been reading your papers, but seeing how they come together, it's quite enlightening. And I'm always thrilled to hear your stories. And I'm sure that many of the audience are feeling the same. Thank you, and thanks again for being with us.
Thank you very much for welcome questions. Yeah.